Okay, in Chapter 5, we're going to start talking about protein function. So in Chapter 4, we spent a lot of time talking about the three-dimensional shapes of protein and different types of structure, primary, secondary, tertiary, and even quaternary. And now we're going to see how these proteins function inside of an organism. And there are two main types of protein function. There are the enzymes, whose job it is to catalyze chemical reactions of specific substrates. So we're talking about making the chemical reactions in our body go at a rate that is fast enough to sustain life, and they're very specific in terms of the chemistry that they do. And then we have proteins whose job is some sort of ligand binding. So ligand binding means that that particular protein binds some molecule. It could be a small molecule. It could be another small peptide or a protein. And in this function, that protein serves for transport of that particular binding partner to some part of the cell or some part of the body, or it might be used in something such as cell signaling, so a way that the outside of the cell communicates with the inside of the cell. Both types of these proteins, enzymes and those that bind ligands, are highly specific, right? So again, they don't just catalyze any chemical reaction or bind any ligand. And in this chapter, we're going to focus primarily on the ligand binding proteins. And in chapter 6, we'll move on to enzymes. And most of the knowledge that we have about ligand binding proteins comes from our studies of myoglobin, which I'm going to abbreviate Mb, and hemoglobin, which I'm going to abbreviate HB. And myoglobin and hemoglobin are both oxygen-binding proteins in the body. Myoglobin functions in the muscle as an oxygen storage protein, whereas hemoglobin functions in the bloodstream for oxygen transport. And we're going to look at how the structures of these two molecules are both the same and different. So in terms of their ability to, which gives rise to their ability to bind oxygen, but the differences lead to their differences in ability to bind oxygen, and hence their different functions in the body. So we'll start with a discussion of myoglobin. So the structure, the crystal structure of myoglobin has been around since the 50s. It has 153 residues, and the protein itself is 70% helical. So it's got a lot of alpha helical structure. The interesting thing about myoglobin and hemoglobin, for that matter, is the presence of a heme group. So if you look at these two structures, you can see that both myoglobin and hemoglobin have this high degree of helical structure. And then you can see here in red the heme group. So this is the prosthetic group, and this is the location of where oxygen binding takes place. So here it is up close and personal. So the heme group consists of a porphyrin ring with an iron 2+ in the center. So the porphyrin ring is this part around here. And it's planar, so we're talking about a really flat chemical structure here. And the iron 2+ is in the middle. So this is going to be the site of oxygen bonding. And what you should see here are 6 coordination bonds. So 1, 2, 3, 4. So 4 coordinating bonds to the iron 2+ and the ring, the porphyrin ring. And then we've got a couple more that you can't see in this picture. So let's get another one here. Okay, so here's the ring. And then we've got 2 coordinating bonds out of plane. One that's responsible for coordinating to the bound O2 molecule, and one that coordinates to a histidine in the rest of the protein structure. And so this binding of oxygen is going to be a reversible process, and it's important, though, that we have this oxygen binding to iron 2+ take place inside this porphyrin ring. If the iron was instead free, Fe2+, if it was just floating around in the body, a free heme would get oxidized to Fe3+ in the presence of that oxygen molecule, and it would just stay that way. It would never go back. So once it got oxidized, it would be done. So having the heme be part of this protein is a way that we can have reversible oxygen binding. So that means, for example, You could bind an oxygen to myoglobin, release it later in the bloodstream, or excuse me, in the muscle, and bind another oxygen at a later time. So it's a reversible process. Now, hemoglobin, on the other hand, even though structurally it has a high degree of helicity, much like myoglobin does, hemoglobin, on the other hand, is tetrameric. That means it takes 4 hemoglobin molecules to come together to make a functional hemoglobin. So we've got then quaternary, I'll put a little quaternary structure that's taking place here. So since it's tetrameric, that means we have 4 hemes in functional hemoglobin, and that's 1 per subunit. So 1 for each individual hemoglobin molecule. So let's take a look here. So here's hemoglobin, its quaternary structure, and what we can see is we've got 2 alpha chains. So those are shown in gray here, each with a heme. And then we've also got these 2 beta chains. So they're all, both the alpha and the beta chains have similar number of residues, 141 and 146 amino acids respectively. And they both have high degrees of helical structure, but they're slightly different in their amino acid sequence. And when these chains, these alpha and beta chains come together, we have some strong non-covalent interactions that take place between those subunits. So looking here at the interfaces between these subunits, we've got a lot of non-covalent interactions. And in particular, we're going to see quite a few hydrogen bonds and ionic interactions that are going to be important later in modulating the binding of oxygen to hemoglobin. Now, because of this quaternary structure, myoglobin and hemoglobin have different mechanisms of oxygen binding, and hence they have different biological functions. And I'm going to get back to the difference between this T state and R state for hemoglobin later on. But it's important to know that oxygen binding is a reversible mechanism. So although they bind it and release it differently, both myoglobin and hemoglobin have the ability to grab oxygen and let it go, but just to different extents. And in our next lecture video, what we'll do is we'll talk about how we look at binding for both myoglobin and hemoglobin, and how we look at some of those association constants for oxygen.